mutation. I wanted to introduce our speaker for this next session, Dr. Thomas Blanchard. He's the director of the University of Maryland Brain and Tissue Bank, as well as the NIH NeuroBioBank. He's here to discuss, biobanking resources the organization can provide to VCP researchers. We have worked together. We probably talked a year or two ago.
Sure.
Time's flying. We're having so much fun. Working together on how to work with patients on donating their brains and biosamples to the biobank. And so we've actually established VCP protocols to do that, so that the patients, when they call in, the source or site knows what, samples to pull, for the patients, and having them consent before the time of death. So a little morbid, but we'll, what I wanted, this is a great opportunity for everybody on to learn what resources, Dr. Blanchard, you and your team have. So I welcome you to the session. And if you want, I'll go ahead and mute. And if you want to try to share your screen real quick.
Bank can be of help and research towards helping those with VCP. I'm going to speak today about the NIH NeuroBioBank, as well as the University of Maryland's Brain and Tissue Bank specifically. And I'm going to talk at first about it from the benefits from a researcher's perspective, and then secondly, from a potential donor's perspective. So the NeuroBioBank is composed of a federation of 6 brain banks, but they weren't all formed as the NeuroBioBank.
Hold on, Tom, we don't have your slides showing.
I'm sorry.
I tried to share them, but I'm going to let you do it so that way you can transition.
Okay.
Hit that little button with the stripe through it.
On the video monitor stripe?
Yeah. Actually, all right, let me share your slides, and you just tell me when to transition. Okay. That way we don't. Technology, we got to love it, don't we?
Mm-hmm.
Okay. Just tell me when.
I don't see them now. Oh, wait a minute. Yeah, let me get rid of that. Okay. Yes. No, back one, please, Nathan.
Mm-hmm.
So the NeuroBioBank was composed of 6 banks that had already existed. And so they all had their own missions and their own individual character. And the Maryland Brain and Tissue Bank was founded by Ron Zielke, or initiated by Ron Zielke back in 1991. It was he had to start this from the ground up. There was no template for building a brain bank at the time. He had to develop protocols, networks, infrastructure, regulations. It was quite a task, but I think he built something that is somewhat of a flagship in the brain bank industry, and he loved it right up until the time he retired it in 2017, and he considered it his pride and joy. Next slide, please. And this was a quote he frequently posted when he presented posters or when he went to meetings. "Some diseases are uniquely human and ultimately require study of human tissue," in an effort to encourage people to interact with the bank and see what we had to offer. Next slide, please, Nathan. So the mission of the bank, as founded, the Maryland Bank, was to provide a mechanism by which families can donate tissue for research, to provide researchers access to human tissue to better understand human diseases, and to accelerate research to improve the treatment, cure, and prevention of developmental and neurologic disorders. This was a mission initially and still funded primarily by the NICHD, and as such, it was devoted to infants and children, one of the few banks, I think, to target that population. It also was very broad in the types of diseases that were accepted into the bank. It wasn't just donors with clinical brain diagnosis. It was children that had any type of developmental or muscular or metabolic disorder. It was quite broad. Next slide, please. It was funded, as I said, in 1991. We're in our 6th competitive renewal for a total since 1991 of over $40 million, including our current budget, which is over $2 million this year. We have received additional funding through some private foundations, including Autism Research Institute and the Blazeman Foundation for ALS. And we really enjoy those interactions with the foundations. And these kinds of things allow us to increase our exposure and outreach to these specific communities. Next slide, please. So when a brain is received by the Brain and Tissue Bank, it is received, normally it's received fresh. And this is going to be the same for all the banks that are in the NeuroBioBank. It is typical that the right hemisphere will end up fixed in formalin and the left hemisphere will end up frozen. And so the right hemisphere, the hemispheres are separated. The right hemisphere is fixed for several weeks. And then samples are taken from all over the hemisphere, depending on the disease that the donor had, the age of the donor. Sample sites might range from 12 to 17 different sites, and depending on the disease or the age, the neuropathologist will request specific immunohistochemical staining to help characterize and produce a neuropathology report. Once those biopsies are taken for histology, the remaining hemisphere is sliced into coronal slabs. The left hemisphere, which will be frozen, is used for quality control. So biopsies are taken from the prefrontal cortex as well as the cerebellum and processed to get a number of different parameters, including pH, RNA, DNA purification values, but most importantly is the RIN value, the RNA integrity number. A lot of researchers, when they're asking for brains, are asking for ones with a low postmortem interval, the time between death and preservation of the brain. But a much better indicator of the integrity of the brain is the RIN value. So the better the integrity of the RNA, the belief is the better the integrity of the tissue that it came from. And so it's very, very common to have a PMI of over 24 hours and still have a very good RIN value. We look for RIN values that are above 6, preferably 7 or 8 or even 9. It turns out that what is more important than time, or at least as important in time, at least up to a point, is how fast that brain was cooled and removed from the body and then kept cold. Once those biopsies are taken, the remaining hemisphere again is sliced into coronal slabs for long-term storage. Next slide, please. This is just a quick diagram. You can look over on the right at the way these hemispheres are sliced. They're all sliced the same. And you can see this one is numbered from 1 to the low 20s in odd numbers. And so the left hemisphere will receive odd numbers. The right hemisphere will receive even numbers so that we can tell by the labels on the bags they end up in immediately which hemisphere it was from and how it was preserved. When someone requests a specific region of the brain, we do not send them an entire coronal slab. We'll pull the correct slab or slabs from the freezer or fixation and dissect out the relevant portions of the tissue and then put the slab back for further use. You can imagine that in that way, a single brain can benefit hundreds and possibly even thousands of researchers, as most are asking for milligram quantities of tissue or isolated DNA or RNA. Next slide, please. And so the ways in which tissues are being used that are requested from the NeuroBioBank are vast. We will loan out coronal slabs and sometimes even hemispheres when planned in advance for imaging, for MRI imaging and other types of imaging. They're often used for histology, single cell analysis. Our bank will occasionally isolate skin biopsies from donors to grow out fibroblast cell culture lines, which we keep banked. People are obviously doing protein studies as well as receptor studies, looking at metabolites. And of course, more recently, the use of the tissue to examine gene expression and genomics is becoming more and more common. In addition to all those experimental methods, we also make medical data available. Any medical records or neuropathology reports or autopsy reports that we get on a case is readily available to a researcher upon request after it's been redacted. Next slide, please. This is just a quick shot of a portion of the bank. We have a dedicated area in the basement of one of our buildings. It was remodeled by the institution specifically for the brain bank. One of the biggest nightmares for, I think, any type of tissue bank is that a freezer will fail on Friday evening and will not be discovered until Monday morning, and you'll get a loss of tissue. And this has happened to some banks. And so most banks have a probe within the freezer that sends a signal to a central location to monitor the temperature, and if the temperatures rise above a certain level, one of us will be contacted at night to address the problem. In our bank, we also have a secondary backup system. Each freezer has a liquid CO2 tank attached, and if the temperature rises above a certain degree, the contents of the CO2 tank are dumped into the freezer, which causes supercooling and gives us an extra few hours to respond. Next slide, please. This is a quick overview of the footprint of our space. We have approximately 45 freezers at this point, in addition to quarters for the fixed tissue. Plenty of room to grow, though, as the university is renovating the room right next door to ours, and we hope within the next few years to double our space. So plenty of room to grow. Next slide, please. Originally, as I said, we were focused on childhood diseases, neurodevelopmental diseases, and our primary recoveries were in things like autism spectrum disease, Down syndrome, Fragile X, Rett syndrome, a wide variety of disorders. And I highlight this to make the point that our bank is very broad in the types of cases that we will recover. And that is not the case in a lot of brain banks, and it's not even the case in amongst all of the banks within the NeuroBioBank. Next slide, please. So the NIH NeuroBioBank was actually started in 2013, and it included 5 banks, but not the University of Maryland. The University of Maryland was invited to join, was requested to join in 2014. This is right from the NeuroBioBank's website. Specimens that span neurological, neuropsychiatric, and neurodevelopmental diseases and disorders, the NBB serves as a central point of access to the world-class collection of our 6 biorepositories. And so that statement, it seems pretty broad, but the 5 banks that were originally part of the NeuroBioBank really had a focus, and still do, on collecting brains from adult cases, but not just adult cases, those cases that had a clinical brain diagnosis. If a donor had some type of disease that was not a clinical brain diagnosis, what is considered a clinical brain diagnosis, like Alzheimer's disease or frontotemporal dementia, then for the purposes of the brain, it was considered a unaffected control. Next slide, please. Once we joined the NeuroBioBank, we readily adopted the mission of expanding the cases we collected. This data is from our previous contract period from 2014 to 2019. We're right in the middle of our current contract period, so this is the most recent data set that I have. And you can see that we, for recovered cases within that time frame, we collected a lot of ALS cases, Parkinson's disease, Alzheimer's disease, several types of dementias. And so we were able to broaden our reach beyond the pediatrics cases and beyond those cases that uniquely affect children to include adults with diseases of many types. Next slide, please. These are the 6 banks that make up the NeuroBioBank network. There's the Maryland Bank, the University of Miami Brain Endowment Bank, the Harvard Brain Tissue Research Center, the Mount Sinai Brain Bank, the UCLA Human Brain and Spinal Fluid Resource Center, and the University of Pittsburgh Brain Tissue Donation Program. We're all part of a federation within the NeuroBioBank. But we still, because these banks were pre-existing when they joined the NeuroBioBank, still have their unique characters, and they still have a degree of autonomy. We meet together regularly. We try to unify and make our protocols as consistent as possible. We take direction from the administrators at the NIH, but we still have a lot of discretion with regards to the cases that we will recover and how we handle the tissue. Next slide, please. Obtaining brain tissue from the NeuroBioBank has many benefits. Not all brain banks are created equal. And one of the first benefits is the cost issue, and that is that there are no costs. If you're a researcher requesting tissue from the NeuroBioBank, whether it's a single case or an order for 200 different cases, there will be no charge other than shipping. If you hope to donate tissue, there are no charges as a donor, but I will talk about that a little bit later. So this is just a remarkable benefit to researchers, and it's not just available to those in the United States. We and the other banks ship tissues throughout the world. The inventory is another big benefit. All 6 banks' inventory is combined on one big database present and accessible on the NeuroBioBank website. It represents well over 10,000 cases and possibly 15,000 cases. And that inventory increases by about 600 to 700 cases per year. You also get, if you so desire, a wealth of information. As I mentioned, medical records, neuropathology reports, toxicology, serology, autopsy reports, and questionnaires are available to you upon request after they've been redacted. Not every case will have all of these types of information, but whatever information we can get on a case can be made available to you. The interface is also quite strong. The searchable database on the NeuroBioBank website is quite remarkable, and you can search simultaneously all 6 databases. You can choose to only search tissues from a single bank or all banks. You can select by disease. You can select on age. It's just a very powerful interface tool. And the service, I believe, is also quite strong. Whether you're dealing with the NIH specifically when trying to place a request, there's a very responsive staff there that can address your questions and help walk you through the process if need be, or even help you work through your application. And once a request is accepted and farmed out to a bank or several banks, the service you'll get from the individual banks is also quite high. At that point, you can freely interact with the technicians at the banks that are handling your case, and it really becomes teamwork. It can, if needed, become teamwork at that point as you address questions that come up about tissue availability and maybe replacing some cases with others. Next slide, please. Requesting tissue from the NBB is spelled out here. This is also a screenshot from part of their website. It's quite simple and straightforward, I believe. You create an account, which only takes a couple of minutes. You can review key documents such as standard operating procedures and such, and then you can get right into browsing the inventory. If you find the cases you're interested in, you can then proceed to make a request. It will require uploading a PDF of your CV, as well as initiating a material transfer agreement, which can also be done or is facilitated by the website itself. Your request will be reviewed by the NIH and the banks, and as approved, it will get farmed out to the individual banks to fill the order. Once fulfilled, you won't interact with the banks anymore, but you will have to provide a progress report to the NIH. I believe it's a year or so after receiving your tissue. Next slide, please. This is a list, and it changes a little bit year by year, but this is a list of the top 20 diseases that are requested from the NeuroBioBank every year. I highlighted the ones in blue just to indicate the demand for the dementia diseases. And yet, despite that interest, a lot of rare diseases and a lot of childhood diseases still make up a significant portion of the most requested diseases from the NeuroBioBank. Next slide, please. So I'd like to now talk about our bank specifically and why the VCP community can benefit from interacting more specifically with the University of Maryland Brain and Tissue Bank. Next slide, please. So as I said, we are quite adept at recovering rare diseases, rare disorders, and we continue to do so. And it's an integral part of our mission. Because the NICHD continues to fund us, we remained, it is essential for us to continue a broader mission than the other 5 banks, which means collecting not just from people with rare diseases, but collecting from people that don't necessarily have a clinical brain disorder. Next slide, please. This is just a snapshot from 1 year indicating the rare diseases we took in that 1 year. You can see the breadth of those diseases. I also put this up to indicate the range of ages that we collected from in that year, from less than 1 year old to between 60 and 69 years old. Next slide, please. So there are several reasons I believe that Maryland is the perfect bank for you to be working with. One of those reasons is that we collect more than just the brain. And so the other 5 banks are as good as anybody at collecting the brain, cerebral spinal fluid, serum, maybe a muscle sample. But we are the only bank within the NeuroBioBank that will make a concerted effort to recover tissue in addition to the brain. This graph shows between 2014 and 2018, the percentage of samples that we sent out that are non-brain. And you can see that percentage ranges from 17% to 44% of the samples we send out are actually non-brain tissue. If you look at the list on the right-hand side, you can see the breadth of the tissues that we collect. A lot of internal organs, muscle, reproductive organs, alimentary tract. I don't mean to suggest that these tissues are collected from every donor. And there's no donor from which all these tissues are collected. But we tend to get a lot of systemic tissues from cases that come from our medical examiner's office. And when we do a remote recovery for a disease which we think it benefits to collect systemic tissue, we will negotiate with the recovery technician to recover systemic tissue. And if we can get them to do it within a reasonable price, we will get the systemic tissue. Next slide, please. Another reason Maryland, I think, remains a very strong bank is the number of control cases that we get. Because we have so many controls and are able to get unaffected control donors, we are actually involved in filling the lion's share of requests that come to the NeuroBioBank each year, whether that is filling the request exclusively or participating with others in a team method to fill a request. So we collect systemic tissue when we can, and we have a lot of controlled tissue to complement researchers' needs. Next slide, please. Another reason is that we've already started working with the VCP community, as Nathan was speaking about in the introduction. We've had several meetings, and we have cases that were recovered as VCP donors by Dr. Kimonis in our bank at the moment. And we look forward to continuing working with her to increase the size of our inventory. Our inventory also includes cases with diagnoses such as Paget's disease of the bone, inclusion body myopathy, as well as frontotemporal dementia. Next slide, please. So as I said, we do have some cases that we would consider VCP candidates or may have actually been genotyped for having the mutation. But they're not listed. If you go to the NeuroBioBank website today and search for them, you won't find them. With Nathan and VCP Cure's encouragement, I have tried to address that with the NeuroBioBank, and that problem has been solved. The issue is that, as I said before, the NeuroBioBank is really concerned with donor cases that have a clinical brain diagnosis. VCP is not considered to be a clinical brain disease. And if the donor didn't have frontotemporal dementia, it wouldn't be collected as such. The NeuroBioBank database will only allow us to upload cases that have a clinical brain diagnosis. A control case, so say somebody donates that has Crohn's disease, that's a non-clinical brain diagnosis. But for the sake of the brain, that case will be considered an unaffected control. And then having filled in that field, it'll be allowed to be uploaded to the database. The trouble is, if we categorize these VCP cases as unaffected controls as far as the brain goes, those cases will be used as controls for other people's requests that are unrelated to VCP. And when a VCP researcher makes a request, we'll find that those cases are depleted. So we've worked with a subcommittee at the NeuroBioBank on this. And we have this and the system there is based on the ICD-10 code tree. And we have decided to place this under the category of genetic carrier of other diseases as a clinical brain diagnosis. If you're searching the database in the future, and I hope by October these cases will be uploaded, the uploads occur 4 times a year, you would search for the following: VCP/IBM-PFD, and you would be able to find any case that we've uploaded as a VCP case based on the genetics. Next slide, please. All right, so what about donating to the tissue bank? Donating should be straightforward. This is a screenshot of our website. You can go to our website, you push that, what should be a red button there that says register, and you will find a phone number or a link where you can contact one of our project coordinators and begin the registration process. Next slide, please. The benefits to donation, I think, are strong. There's no cost to the donor or the donor's next of kin ever. The bank makes all arrangements for tissue recovery, and that includes transport for the donor. So if the donor is at a place where recovery can't be made, then the bank will arrange to have the body transported to a facility or a funeral home where that recovery can be made, and then transport the body back to where it needs to be for the family's sake. No charge to the family. Recovery, as I said, can include the brain as well as systemic tissue. And although pre-registration is optimal, donation can be arranged post-mortem as long as the bank is contacted in a timely fashion after death that gives us enough time to recruit an autopsy technician after we get consent. Next slide, please. Okay, if you go to the NeuroBioBank directly to register as a donor, you will be redirected to something called the Brain Donor Project. The Brain Donor Project is a foundation, nonprofit foundation that was established for the sole purpose of helping the NeuroBioBank register people to donate their brain. And so if you go to the NeuroBioBank website, you're going to be directed to push this button for the Brain Donor Project. Next slide, please. So you will call somebody at the Brain Donor Project, and they will get some preliminary information from you. And based on where you live in the country, your case will be directed to one of the 6 banks. You'll be contacted by that bank, and then the registration process will begin. The pre-registration process will begin. Next slide, please. So the problem with that from the VCP community is that if you look at the states in yellow, this is the University of Maryland catchment area as far as the Brain Donor Project is concerned. And so if you live in one of those states and you try to register through the NeuroBioBank/Brain Donor Project, you will be directed to our bank, and we will know how to handle a VCP case. If you live somewhere else, you won't necessarily be directed to Maryland. Next slide, please. But this slide is meant to illustrate that we recover from anywhere in the United States. The states in yellow represent states and the number of recoveries that were made during the previous contract period. Next slide, please. And these numbers represent the number of people that are pre-registered to donate specifically to the University of Maryland Brain and Tissue Bank. And so we have a network of autopsy technicians throughout the country, and we can recover from anywhere in the country. Next slide, please. And so what is the best way for a VCP donor to register to make sure that upon recovery, things will be done for the benefit of the VCP research community? One is you can continue, or you can use the Brain Donor Project to register. But it would be important to emphasize the fact that you're calling as a VCP disease donor. And if the Brain Donor Project doesn't already appreciate the significance of that, you can ask for your case to be directed to the University of Maryland Brain and Tissue Bank. They will willingly honor that request. Or you can contact the University of Maryland Brain and Tissue Bank directly using our website or by telephone. Next slide, please. Registration process requires a lot of shuffling of documents back and forth. Step one, you can call any time of the day or night or weekend. Off hours, you'll get an answering service, but that message will be forwarded to one of our project coordinators, and then you will be called back. A registration packet will be sent to you, either online or through the mail, which receives the consent document, the HIPAA document, and a questionnaire. And we have to receive that back before we can finish the screening process and have you pre-registered. And once you're pre-registered, we won't talk to each other again until the donor is near death or death has occurred, in which case you'll call us and we will set things in motion. When someone dies, and this is put in motion post-mortem, this really is time sensitive. We have to get these documents to the next of kin and receive them back before we can initiate things. But if the phone call comes in quick enough, we can usually have that brain recovered within 24 hours. Next slide, please. So this slide is just to represent the different sources of our tissue. The Brain Donor Project didn't come into existence until 2016. And since 2016, the Brain Donor Project referrals are represented in blue here. You can see the number of cases that are referred to us from the Brain Donor Project has increased, and I don't have the data here, but it has leveled off. All those other colors represent people that have registered with Maryland directly, or have been referred to us by collaborators directly. And so there is no wrong way to register for brain donation. The important part is that you get registered. Next slide, please. So I'm over time already. I just want to highlight the people that do the work, and particularly the project coordinators over on the left, Anthony Weldon and Grace Flannery. If you call to make a donation, you'll likely talk to one of those two. And in the middle at the top, Lexi Alexander and Robert Johnson. If you're making a tissue request, you will undoubtedly speak and work with one of those two. Next slide, please. And I'll wrap it up there, and thank you for your attention. And I leave you my phone number and email address, and I would be more than happy to speak with anybody if they want to learn more about how we might be able to help and actually teach me more about VCP. Thank you.
Juggling lots of things. So thank you. What questions do we have right now?
I don't see any.
Yeah, don't see any. I know we've got, looks like 27 people on right now. Does anybody have any questions for Dr. Blanchard? Thank you. That I wish I'd had that presentation when we first started. Maybe we incorporated that stuff in the Brain Donor Project. That was good. That was great. And thank you for advocating on the VCP piece, you know, to get that captured. But yeah, I think the takeaway is we definitely recommend calling Maryland directly versus trying to funnel through the Brain Donor Project. Yeah, and I know Virginia sent you an email, Tom. She just yep. So
I owe you a response, Virginia. Thank you. I was able to download the documents. I appreciate it. I will be in touch.
Yeah, we're all strapped right now. All right, any questions from anybody?
Thank you for listening.
Yeah, no, thank you. This was great, and we'll have a recording of this because I will definitely be using this for the future. So if there's no questions, what we'll do right now is go ahead and close this session. We start at 6:30 with 6 abstract presentations. And so it's going to be really difficult to, the 10 minutes each, we're going to have to change over. You see with the technology, transitioning is a challenge. But we'll go ahead and close out this session so you can leave right now. Everybody has a little break right now. I know Thomas, you have a meeting to go to, so we'll get you out of here early, but we will see everybody at 6:30 in the next session. Then thank you very much. This is closed. Thank you.